When and why should I consider flow cytometry? And what kind of information can flow cytometry experiments give me that Western blots can't? I'm Sarah, development scientist at Cell Signaling Technology, and this is CST Tech Tips. Western blotting is used to detect proteins and analyze their expression using antibodies. But Western blotting does have its limitations. Flow cytometry also employs antibodies, but it uses fluorescence to detect and analyze protein expression on a cell-by-cell basis. Here are 4 situations where it can be worth going with flow over Western blot. Situation 1. You want to analyze multiple targets in the same sample in a shorter time. Multiple targets means using multiple antibodies. To do this with Westerns, it's possible to strip and reprobe the blot or set up multiple blots. But reprobing adds a lot more time to your workflow, and running blots in parallel requires more lysate. Multiplexing antibodies with flow saves time, and you can get more data from your samples. Situation 2. You want to analyze specific cell populations or subpopulations. This could apply if your input material contains cell types, for example, tissue or immunological samples. Flow cytometry allows phenotyping of populations and subpopulations by employing antibodies to proteins selectively expressed in each cell type. Because of this, flow cytometry is widely used in studies of immunology, cancer biology, developmental biology, and more. Situation 3. You want more sensitive detection of intracellular signaling events. Let's say you're measuring a signaling response to a stimulus, but only a portion of the cells in your sample are responding. This is not uncommon for many signaling readouts. In Western blot, lysates are pooled into one sample, so your signal from the responding cells will be effectively diluted out. But in flow cytometry, analysis is conducted on a single cell basis, enabling you to detect and quantify these rare signaling events and define the cell types in which they occur. Situation 4. You want to analyze multiple readouts and look for correlation between them. For example, if your hypothesis is that 2 subpopulations will respond differently to a treatment, you can design an antibody panel including antibodies to multiple phenotypic surface markers to gate your populations, and antibodies to evaluate expression or activation of signaling proteins. While multiplexing may require optimization, the benefits of this multi-parametric analysis can be a very powerful tool to gain biological insights. Once you've decided to add flow to your experimental toolbox, here are 2 steps to take next. First, you'll have to arrange access to a flow cytometer, either through your institution's core facility or a collaborator. Second is to start designing your antibody panel, keeping in mind the antibody you used in your Westerns may not perform in flow. So when you're selecting antibodies and antibody conjugates, make sure to look for validation data performed specifically in flow cytometry. If you found this video helpful, hit the like and subscribe buttons and share with your colleagues. You can find application-specific protocols under the antibody product pages on cellsignal.com. Anytime you have a question about an antibody or a protocol, you can ask one of our scientists at cellsignal.com/support. Good luck with your experiments.